Movement highways connect multiple brain structures. The main hubs of movement neurocircuitry are the cerebral cortex, basal ganglia, thalamus, and cerebellum. The motor cortex is the primary movement hub of the cerebral cortex, which receives and sends signals that result in selection, preparation for, and control of voluntary movements. The basal ganglia are subcortical masses of cell bodies organized into structures consisting of the striatum, substantia nigra, subthalamic nucleus, and globus pallidus. This collective hub sits at the base of the forebrain and on top of the midbrain, and interconnects with the cerebral cortex, thalamus, limbic system, and brainstem to control movement, cognition, and emotions. The striatum is comprised of dorsal and ventral segments. The dorsal striatum plays a key role in the regulation of normal movement, whereas the ventral striatum regulates reward and reinforcement. The substantia nigra is located in the midbrain of the brainstem. It is a primary source of dopamine in the brain, including to the striatum for modulating movement. The globus pallidus is located medial to the striatum and is subdivided into two parts, internal and external. Its role in movement is to inhibit the subthalamic nucleus and thalamus. The subthalamic nucleus is located inferior to the thalamus and is functionally connected to the globus pallidus. The thalamus, located medial to the striatum, is not part of the basal ganglia. It translates movement-related information received from the basal ganglia to the cortex. The cerebellum is located in the hindbrain, and like the basal ganglia, it forms pathways to the cortex via the thalamus to modulate voluntary movement. Unlike the inhibitory output of the basal ganglia, the cerebellar output is excitatory. The cerebellum is located in the hindbrain, and like the basal ganglia, it forms pathways to the cortex via the thalamus to modulate voluntary movement. Unlike the inhibitory output of the basal ganglia, the cerebellar output is excitatory. Which 5 structures comprise the basal ganglia? Select which part of the striatum plays a key role. Select which part of the striatum. Module 2, Neurocircuitry, Movement Highways in the Brain. The basal ganglia perform multiple functions. Basal ganglia nuclei form circuits with the cortex and thalamus to control movement, cognition, and limbic functions. The circuits serving these functions are largely segregated and parallel within this shared anatomy. The motor circuit controls movement and is comprised of the motor cortex, specifically the supplementary motor area, the dorsal striatum, the globus pallidus, substantia nigra, subthalamic nucleus, and thalamus. The cognitive circuit is comprised of the dorsolateral prefrontal and lateral orbitofrontal cortical areas, the dorsomedial striatum, globus pallidus interna, substantia nigra, and thalamus. The limbic circuit is comprised of the anterior cingulate and prefrontal cortices, ventral striatum, ventral pallidum, and the thalamus. Inputs to the basal ganglia are also received from the amygdala and hippocampus. Module 3. Neurotransmitters. Stop and go signals for movement. The nervous system controls movement and other functions through coordinated actions of several neurotransmitters. Key neurotransmitters involved in movement include glutamate, GABA, dopamine, and acetylcholine. Glutamate is the main excitatory neurotransmitter. The basal ganglia receive extensive excitatory glutamatergic input from the cerebral cortex. Gamma-aminobutyric acid, or GABA, is the main inhibitory neurotransmitter. It is the primary outgoing signal from the basal ganglia to the thalamus. In addition to glutamate and GABA, there are numerous modulatory neurotransmitters. The key ones are dopamine and acetylcholine that modify or focus the signals in the basal ganglia pathways for normal movement. Dopamine, produced by neurons in the substantia nigra pars compacta, signals to the dorsal striatum to regulate excitatory and inhibitory pathways that contribute to movement control. Within the dorsal striatum, dopaminergic neurons act upon medium spiny neurons to regulate movement. In general, dopamine provides a go signal for movement. Dopamine is released via synaptic vesicles to freely associate and disassociate from postsynaptic receptors. Depending on the receptor type, dopamine can either act as an excitatory or an inhibitory neurotransmitter. Dopamine imbalance in the basal ganglia results in various involuntary hyperkinetic movement disorders, such as Huntington's disease chorea and tardive dyskinesia, and hypokinetic movement disorders like Parkinson's disease. Acetylcholine is produced in a population of striatal interneurons. It is also modulatory, like dopamine, where its function depends on the receptor type, but generally provides a stop signal to movement. Serotonin and norepinephrine are lesser understood neurotransmitters in the basal ganglia network that appear to modulate dopaminergic control for normal movement. The ultimate actions of these neurotransmitters depend on the receptor type and location that they engage, and other adjacent neurotransmitter inputs. Which neurotransmitter is the primary outgoing signal from the basal ganglia to the thalamus? Which neurotransmitter is the primary outgoing signal from the cerebral cortex to the basal ganglia? Which dopamine receptor acts as an excitatory neurotransmitter? Which dopamine receptor acts as an inhibitory neurotransmitter? Module 4. Normal movements. Coordinated commute. Reaching for and grabbing a glass can seem like a simple task, but within the brain, multiple pathways work together to produce voluntary movements only when and where desired. In short, initiating and carrying out the desired movement is controlled by the direct pathway. At the same time, the indirect pathway is working to stop any unwanted movements from occurring, like your hand opening when you don't want it to open. Together, the balance of these pathways enable control of our movements. Now, let's take a closer look at what goes on within these pathways. Regulation of voluntary muscle movement starts in the cortex of the brain. The motor cortex sends glutamatergic, excitatory signals to the dorsal striatum, and from the striatum, GABAergic, inhibitory signals project to the internal globus pallidus as part of the so-called direct pathway, and to the external globus pallidus as part of the indirect pathway. Normally, at rest, the basal ganglia continuously inhibit the thalamus to suppress random movements. This occurs because the GABAergic neurons of the internal globus pallidus actively inhibit the thalamus. The direct and indirect pathways have opposing effects on this inhibitory signal. The balance of the direct and indirect pathways within the dorsal striatum dictates which movements are carried out. Signaling from the direct pathway results in a go signal for desired movements. This happens through brief disinhibition of the thalamus, allowing excitatory signals to be sent to the cortex to initiate movement. The indirect pathway is the opposite. Through an additional synapse, the indirect pathway results in a no-go signal for movement. This occurs through increased inhibition of the thalamus, effectively stopping signals to the cortex and preventing movement. When a movement is desired, for example, raising a glass of water, the motor cortex excites the dorsal striatum. The dorsal striatum processes this signal by way of the direct and indirect pathways to generate the desired movement. To generate the go signal, the direct pathway inhibitory medium spiny neuron sends GABA signals through the globus pallidus internal or substantia nigra pars reticulata, ultimately allowing the thalamus to excite the motor cortex to produce movement. Thus, through release of this normally inhibitory signal to the thalamus, the direct pathway enables movement. In contrast, the indirect pathway prevents competing or unwanted movements. To generate this stop signal, GABA medium spiny neurons within the dorsal striatum inhibits the external globus pallidus. This, in turn, releases the external globus pallidus inhibition of the subthalamic nucleus, allowing for excitatory glutamate signals to be sent to the internal globus pallidus. As a result of globus pallidus internal activation, inhibition of the thalamus is increased, preventing it from sending signals to the motor cortex. Thus, the indirect pathway results in inhibition of movement. Recall that the dorsal striatum receives dopamine signals from the substantia nigra pars compacta via the nigrostriatal pathway. Within the dorsal striatum, these dopamine signals modulate GABA medium spiny neurons of the direct and indirect pathway to facilitate movement. Medium spiny neurons of the direct pathway express D1 receptors. When dopamine binds to a D1, it increases the sensitivity of the medium spiny neurons and thereby supports the activation of the direct pathway. Overall, dopamine through D1 receptors facilitates the direct pathway's role in producing desired movements. In contrast, medium spiny neurons of the indirect pathway express D2 receptors. When dopamine binds to D2, it decreases the sensitivity of the medium spiny neurons and thereby suppresses the stop signals from the indirect pathway. Thus, dopamine through D2 receptors suppresses activation of the indirect pathway to allow desired movement. Overall, through activation of the direct pathway and inactivation of the indirect pathway, dopamine enables movements. Additional regulation of the direct and indirect pathways occurs via a population of cholinergic interneurons in the dorsal striatum. Opposite to dopamine, acetylcholine signals suppress the go signals of the direct pathway and activate the stop signals of the indirect pathway. Thus, acetylcholine overall suppresses movements. In summary, without the basal ganglia, uncontrolled and competing movements would be the norm. The basal ganglia are like traffic signals, keeping movements in check. Through the balance of dopamine go signals and acetylcholine stop signaling on the direct and indirect pathway neurons, the dorsal striatum introduces gating or traffic control in the basal ganglia to control movements. Select the label of the correct pathway. Which is the direct pathway? Select the label of the correct pathway. Which is the indirect pathway? Select the label. Which pathway enables desired movements? Select the label. Which pathway inhibits undesired movements? Which dopamine receptor activates the direct pathway, facilitating movement? Which dopamine receptor activates the indirect pathway, inhibiting movement? Within the basal ganglia, which neurotransmitter facilitates desired movements? Within the basal ganglia, which neurotransmitter suppresses movements? Module 5. Pathophysiology of abnormal movements. Disrupted commute. Dysfunction of the direct and indirect pathways can result in both hypokinetic and hyperkinetic movement disorders. The cardinal feature of hypokinetic disorders is bradykinesia, or a decrease in overall movements. For hyperkinetic disorders, the predominant feature is chorea, or excessive involuntary movements that appear as randomly flowing, jerky movements. The hypokinetic disorder, Parkinson's disease, is characterized by bradykinesia, rigidity, and resting tremors. It is caused by the loss of dopaminergic neurons in the substantia nigra pars compacta. This, in turn, is postulated to cause an imbalance between acetylcholine and dopamine signaling. Decreased dopamine signaling leads to an overactive indirect pathway, or too much stop signal, and underactive direct pathway, or too little go signal. Thus, excessive inhibition of the thalamus by the indirect pathway leads to overall decreased movement, or bradykinesia, the hallmark feature of Parkinson's disease. Drug-induced Parkinsonism is also characterized by bradykinesia, rigidity, and resting tremors. However, it is caused by medications when there is excessive acute blockade of medium spiny neurons, D2 receptors. This creates too much stop signal from an imbalance of dopamine and acetylcholine signaling in the indirect pathway. Thus, loss of D2 modulation of the indirect pathway results in excessive inhibition of the thalamus, leading to overall decreased movement, or bradykinesia. The hyperkinetic disorder, chorea, is characterized by excessive abnormal involuntary movements that appear as random flowing jerks. In Huntington's disease, chorea manifests as a consequence of early medium spiny neuron loss within the indirect pathway of the dorsal striatum. With less stop signal from the indirect pathway, there is unchecked stimulation of the thalamus, which results in excessive involuntary movements, or chorea. The drug-induced hyperkinetic disorder, tardive dyskinesia, manifests as involuntary movements that are typically stereotypic, choreiform, or dystonic. Tardive dyskinesia classically presents as oral, buccal, lingual movements, but can affect any part of the body. One theory for these drug-induced hyperkinetic movements is chronic D2 receptor blockade leads to D2 receptor upregulation and/or dopamine supersensitivity. This upregulation or supersensitivity may result in too much dopamine inactivation of the indirect pathway or insufficient stop signaling, allowing for unwanted hyperkinetic movements to occur. The hypokinetic disorder, Parkinson's disease, drug in an excess. Module 6, Pharmacotherapy of Abnormal Movements, Overcoming Roadblocks. Aging or damage to the direct and indirect pathways can cause hyperkinetic or hypokinetic movement disorders. Certain medications also can cause these movement disorders. In some cases, the same drugs that induce the movement disorder can also mask it. And, not surprisingly, medications that treat one movement disorder can also cause another. For example, medications used to treat hypokinetic disorders by promoting movement can cause hyperkinetic movements, and vice versa. Antipsychotic medications used to treat psychiatric disorders can cause both hypo and hyperkinetic movement disorders. Antipsychotics can exert their therapeutic effect through D2 receptor blockade within the ventral striatum of the mesolimbic pathway. However, blockade of D2 receptors also occurs on medium spiny neurons of the indirect motor pathway within the dorsal striatum. In the short term, excess blockade of D2 receptors of the indirect pathway produces too much stop signal and drug-induced Parkinsonism. Over time, chronic blockade of D2 receptors in the dorsal striatum can lead to an imbalance of stop and go signals, resulting in tardive dyskinesia. Historically, and less often today, the nonspecific term extrapyramidal symptoms is used as a catch-all to describe what are distinct abnormal drug-induced movements. The imprecise nature of EPS can lead to improper use of pharmacotherapy. For example, M1 receptor blocking anticholinergics may be useful for drug-induced Parkinsonism by decreasing acetylcholine-driven stop signals from the indirect pathway. But this further reduction in stop signals can worsen tardive dyskinesia. This emphasizes the complex interplay of neurotransmitter signaling and pathophysiology of movement disorders. While antipsychotic medications can cause tardive dyskinesia, administration of higher doses can reduce or mask the hyperkinetic symptoms. This demonstrates the complexity of dopaminergic dysfunction. One hypothesis for tardive dyskinesia is an increase in the number or sensitivity of D2 receptors. Higher doses of antipsychotics can antagonize these receptors, temporarily reducing the increased dopaminergic signal underlying the uncontrolled movements. In the long term, this approach may worsen tardive dyskinesia. The only FDA-approved treatments for tardive dyskinesia are the reversible vesicular monoamine transporter 2, VMAT2 inhibitors, deutetrabenazine and valbenazine. VMAT2 is a protein in the presynaptic vesicle membrane that transports monoamines, including dopamine, from the cytosol into the vesicle for storage. When VMAT2 is inhibited, less dopamine is stored in vesicles, resulting in less dopamine being released into the synapse for potential activation of D2 receptors. Less D2-mediated signaling presumably supports normalized function of the indirect pathway, consequently reducing hyperkinetic movements of tardive dyskinesia. VMAT2 inhibitors, tetrabenazine and deutetrabenazine, are also approved in some countries, including the US, for chorea associated with Huntington's disease. Degeneration of the medium spiny neurons of the indirect pathway causes an imbalance, resulting in overactivity of the direct pathway. Reducing dopamine through VMAT2 inhibition is thought to normalize this balance by reducing GO signals from the direct pathway. The balance of stop and go signals from the dorsal striatum is critical for control of movement. Although VMAT2 inhibitors can treat the hyperkinetic symptoms of tardive dyskinesia, they also pose a risk of causing drug-induced Parkinsonism, similar to antipsychotics. This is a result of excessive D2 blockade or presynaptic depletion of dopamine, which causes excess stop signals from the indirect pathway. In contrast to tardive dyskinesia, increasing the antipsychotic or VMAT2 inhibitor dose can make drug-induced Parkinsonism worse. In Parkinson's disease, the loss of dopamine-producing cells within the substantia nigra pars compacta results in bradykinesia. The main way to increase synaptic dopamine is the use of levodopa, a dopamine precursor that is metabolized into dopamine by a naturally occurring enzyme called dopamine decarboxylase. Replacing the deficient dopamine within the dorsal striatum can rebalance the indirect and direct pathways to improve movement production. However, too high of doses of levodopa can induce hyperkinetic movements, called levodopa-induced dyskinesia, or LID. Movement disorders can arise from both pathologic damage and medication-induced changes to the motor pathway signaling. Understanding the underlying neurotransmitter signal changes within the direct and indirect pathways is key to selecting the right treatment. Additionally, The fine balance between too much and too little signaling means medications used to treat hypokinetic disorders by promoting movement can cause hyperkinetic movements, and vice versa. Thus, the focus of treatment of hypo and hyperkinetic disorders is achieving a balance of excitatory and inhibitory signals within the movement pathways. Does the inhibition of VMAT2 result in an increase or decrease in the release of dopamine into the synapse? Which of these two movement disorders fall under the term extrapyramidal symptoms? Anticholinergics may be useful for treating drug-induced Parkinsonism symptoms by decreasing the stop or go signals from the indirect pathway. Which of these medication classes include an indication for the treatment of the tardive dyskinesia?